Non-invasive prenatal screening, or NIPS, by cell-free DNA analysis is the most sensitive screening test available for the common aneuploidies Down syndrome, trisomy 18, and trisomy 13. NIPS test failures, also referred to as no-calls, occur for a variety of reasons, and some laboratories employ a fetal fraction threshold, issuing a test failure for all samples with fetal fraction below a certain percent. Current society guidelines recommend that patients with a test failure be informed of an increased risk for aneuploidy and be offered invasive testing. In this study, we described our clinical experience of the Prequel prenatal screen, which leverages a customized whole genome sequencing approach and does not utilize a fetal fraction threshold. We retrospectively analyzed results from a series of more than 58,000 clinical samples. The NIPS results were then compared to the clinical outcomes, which were requested for all screen-positive results, as well as 10% of randomly selected screen-negative results. We found that 99.9% of patients received a Prequel result the first time, for a test failure rate of just 0.1%. Test performance was consistent with that of the largest and most recent meta-analysis of NIPS performance. Importantly, we looked specifically at performance at low fetal fraction, those below 4%. In this subset of patients, we found that Prequel accurately detected nearly 24 aneuploid pregnancies and provided reassurance to thousands of patients who would have otherwise received test failures if a fetal fraction threshold had been employed. Discordant samples at low fetal fraction were rare. Samples were reanalyzed with an updated proprietary algorithm currently used in our laboratory, which reduced the number of false negatives to 2 cases. This underlines the importance of continuous improvement and customization of our NIPS algorithm. In summary, our clinical experience spanning nearly 60,000 patients shows high accuracy while maintaining a low failure rate of 0.1%. A fetal fraction threshold would have negatively impacted thousands of patients. Failing samples based on fetal fraction is not necessary with our custom NIPS.